We promised we'd wait till 5:05. Ashley, that was spectacularly well timed. I don't know how you managed that, but welcome everybody. Thank you for taking time out of your evening to join us. It's my pleasure and honor and privilege on behalf of ONDRI and on behalf of everybody that's on today to welcome you to what I hope will be a really fun evening. I'm going to start off with just a little bit of housekeeping, the obligatory conference housekeeping items. We are going to be recording, so just give everybody a heads up about that. Everybody is muted. We ask to try to keep on mute during the presentations, but we will have time later on this evening for unmuting and banter, toasting, roasting, or other ridicule that people choose to pursue. I think we're fairly safe with this crowd. But we do want you and encourage you indeed, given the timing, to bring food and drink, wine and cheese, dinner, whatever you like to partake of. Washrooms are just down the hall from wherever you are, I'm pretty sure. So feel free to use them if you need them. We will be showing the video messages. There's a nice collection and collage of brief video messages from friends, colleagues, and collaborators and trainees and graduates that will come a little bit later. And we'll be showing those, and we'll be also taking video messages in if people want to add to that pile to give to Sandra later until Wednesday. I wanted to thank friends and family for joining. So Sandra's family is here, Jeff and Adam, and Peter and Susan. Thank you for joining us. Colleagues past, present, and future, welcome, and thank you for coming. I want to also just thank the ONDRI team, so specifically Ashley and Natalie and Ruth for getting the word out, for doing all the behind the scenes work to make this what will hopefully be a seamless evening and pull together different people's thoughts and the technology that we have going on today. So thank you to our ONDRI team for helping us celebrate and welcome Dr. Black. So I think everybody here knows.
I think I have to share my screen, right? Because I've made some modifications to the talk.
You will have to share your screen.
Yeah.
The excuse for tonight is that we're really celebrating the Lifetime Achievement Award from the Society of Behavioral and Cognitive Neurology, and we want to thank the SBCN for giving us an excuse. In these days, in these times, we all need to celebrate good things and get people together and tell the people that matter to us how much they matter. And so that's what this is. This is not a retirement party. This is not a send-off. This is not a career end. This is a celebration. And so it's a good excuse. And we thank the SBCN for giving us that excuse. And at ONDRI, we decided to seize it and run with it and use it as a chance to to both hear from Sandra, because she has put together really a remarkable talk, but also to have her hear from us, which we don't really get the excuse to do otherwise. So it took COVID and a global disaster and pandemic to get her to stay out of the hospital for any length of time. And it's taken that to get her to allow us to tribute her as well. But we're going to seize the moment and seize the day. So without further ado, Sandra, over to you. Thank you for giving us the talk this evening.
Okay, so I'm very touched by this surprise event, which I didn't quite realize what was happening. I thought I was just giving a little talk, and then it turns out that things have been delivered to our condo, and a whole bunch of people are here that I didn't expect, so I'm delighted. And I'm going to just talk a little bit about this is the formal talk, protein misfolding small vessel disease, but really this is about the Sunnybrook Dementia Study. And I think the award partly reflects the privilege I've had over the many years I've been here to kind of build something that to me was very important. And really the trick is, I think, as an example of research embedded in care, where I was learning from my patients and trying to give back to my patients by giving them the best care that I knew was possible at the time. So to start with, I just wanted to acknowledge my core family, Jeff and Adam. This was at the time of my wheelchair celebration when I was given the wheelchair at the University of Toronto. Below are some more family, my mother, brother, and Susan, and her daughter, who's no longer with us. And here's the newest members of my family. Adam, fast forward a few years, Simon and Anna Maria, and he's the joy of my life right now. So the Sunnybrook Dementia Study. This is multimodal research embedded in care. Started in 1995. I've listed some of the key contributors. And I actually want to dedicate this to the memory of Don Stuss, who was the kind of major co-investigator in that we agreed upon and he developed in collaboration with his fellow scientists at the neuropsych battery that we still use today. And it was very comprehensive and it allowed us to describe the cognitive symptoms of individuals. And we added other measures, and it was very sad to lose Dawn to pancreatic cancer a couple of years ago. That's me and Nick Alexander, who was a very close collaborator of Dawn and did a lot of the work with Dawn on frontal lobe function. So I'm not known for short, brief slides. So I do want to cover quite a lot in this, just to kind of give you the setting, and then I'm going to go into some of the data that was generated in this manner, focused on white matter disease or small vessel disease. This was continuously funded since about 1995 and renewed as a foundation grant to 2024. The trick was that we were able to collaborate with neuroimaging, medical imaging, and medical biophysics in a quantitative MRI sequence that turned out to be gold, because it allowed us to do three-dimensional reconstructions of the brain as well as see the small vessel disease in many subtypes. And it was actually superior in many respects to what came later in projects like the Alzheimer Disease Neuroimaging Initiative, because it allowed us to see more of the small vessel disease. We teamed up with Peter George Hislop for genetics. The neuropsychology was really worked out by Dawn. We had functional scales and something called the neuropsychiatric behavioral inventory, which gave us all sorts of insight into apathy and depression and various kinds of behavior, which is actually very important in the different dementia syndromes. Fast forward a little bit, and we were able to start one of the first kind of networks called the Canadian Partnership for Stroke Recovery. You see that Michael Young is here, so he goes back a long way, where the focus was on motor and cognitive recovery, but with vascular cognitive impairment included, which is often not the case in many such endeavors. And so it was sort of a full-on both motor recovery, gait recovery, cognitive consequences of stroke, and that tradition has been carried on by some of my, I guess I could call them sort of academic progeny, one of whom has been introducing today's session. Over time, we started to foray into new techniques like amyloid PET imaging in white matter disease, which I'm going to talk about more. And this interdisciplinary collaboration was taking a further step forward in something called the Toronto Dementia Research Alliance, where we developed an iPad cognitive assessment tool, where we started to do standard intake letters so that everybody would be assessed in the same way. And that's continued to develop and get to the electronic patient record, not quite there yet. But together, when we work across the city, we see about 2,000 new patients a year. And once we have everybody imaged the same way, and we have all these standardized assessments and blood tests, which are going to tell us if people have Alzheimer's and other pathology, I think we can be really a very influential player in the international scene. But the most important part of what I want to talk about is how this has also not just been partnerships with other academic centers and multidisciplinary scientists and clinicians. It's also that the partners have been families. patients and caregivers. And that included, to the bitter end, because often we were following people because we didn't have a lot we could do, although some symptomatic treatments came along, to their demise. And many individuals were willing to allow us to do the postmortem so that we could learn a lot about what we were actually seeing. And I have to tell you that at the time that we got going, even in the mid-1990s, the disease patterns were just really starting to be identified for subtypes of Alzheimer's disease. Parkinson's disease was known for years, but something called Lewy body disease was only described in 1988 with its very terrifying manifestations of hallucinations and REM sleep disorder, where people act out through dreams, and they can be sort of delirious and confused, and the hallucinations can be more than just visual hallucinations. These are only starting to be described. Frontotemporal degeneration was thought to be something called Pick's disease, described around the same time as Alzheimer described Alzheimer's disease in 1906. But there are other types of what's called tauopathy, because Pick was a type of tauopathy. That's a protein, one of the misfolding proteins, that can cause behavioral changes and especially language decline if it affects especially the left hemisphere. And I'm delighted that my fellowship mentor and great colleague, Dr. Andy Curtis, is here tonight, is actually also, I think, the only other Canadian awardee of this Lifetime Achievement Award. So Andy, I'm so happy and delighted that you've been able to come tonight. And Andy mapped out the different types of primary progressive aphasia so beautifully, just as he had mapped out the different types of aphasia when I was with him in the 1980s, and with his focus on recovery and brain mapping. That is really, that was the tool that I really learned from Andy. And in fact, we had the first MRI scanner in the country at St. Joseph's in London, and Andy will remember that we didn't have access to a CT scan or CT scanner at that time, and so people would come in with an acute stroke syndrome. We'd have to wait a few days to get a CT scan, but we could get an MRI scan, and it was at 0.15 Tesla, a tenth of what the standard scanner is now. And it was like for those of you who know imaging, it was like a diffusion scan. You could see the full stroke. You could see a negative CT and a very big stroke. And so we were sort of ahead of the game when we had access to the MRI. And then we'd send them a couple of days later to the CT scanner down the street, and they would show the of the stroke. So that was amazing. And that's how I started to learn about white matter disease, which I'll show you an example of in a moment. And then there's also vascular cognitive impairment, which has many different phases, as I'll talk about later. So really, I learned almost patient by patient through their narratives and through storytelling, which it was informed by the science, it informed the science, and it was informed by the science, to which we eventually were able to contribute because we could do standardized assessments and begin to see patterns and begin to participate in networks that were trying to come up with consensus criteria for these different dementias, that were trying to begin to put together networks to study, for example, in the ADNI study, mild cognitive impairment, normal controls, and Alzheimer's disease, selected cases. The Ontario Neurodegenerative Research Initiative, which my former doctoral students, Rick Swartz and Mary Marcellus, now co-lead, along with my colleague in Kingston. And then the CCNA, the Canadian Consortium for Neurodegeneration and Aging, which is a Canada-wide network where, again, Mary and I have some leadership roles in that network. There are many networks that, by working together, can create data sets and collaborations that are usually multidisciplinary and can inform the science. This is just a list of some of them. To give you a sense, some are foundations and sources of funding, but the TDRA Alliance is shown here, the Canadian Partnership for Stroke Recovery, ADNI, the Duke Foundation, the VASCOG, the Ontario Neurodegenerative Disease Research Initiative. These are all networks in which we could contribute because we had the patient populations, they were well described, and we could get them in and contribute to this larger set of patient multimodal data sets. So that was sort of the concept and the methodology in a way, and this was the setting, a little different. Now, Michael, compared to 1995 even, when I first came. In fact, when I first came to Sunnybrook, there was a park where there is now the parking lot, excuse me. And it was quite a bit smaller, so there were major expansions that went on over the years. But we were away from the downtown hospital, a former veterans hospital, where I think there was a kind of an obligation and a sense of duty to deliver best care possible to individuals, and the kind of research enterprise arose after maybe 20 years of it being an excellent teaching hospital. And actually, when putting a pitch to one of our very kind donors, when they asked sort of what was different about Sunnybrook compared to other wonderful hospitals at the University of Toronto, I think it was a sense of giving service and needing to serve people that maybe arose from its background as a veterans hospital. So now a bit more about my lab family. You've got several pictures you've been circulating. I apologize for my hoarse voice. Let me just take a drink. Unfortunately, not champagne, just water. So this is one moment in the lab's history of a bunch of people. And here are some photos, snapshots of some of the most important contributors who were my colleagues, fellows, and students. I'm not going to go through all of them, but it was fun just trying to capture their pictures, and they all contributed significantly to the Sunnybrook Dementia Study in ways that I could point out. Because as time went on, we then had these offshoots at 3T, the NITNIC study, the Vascular Brain Health Study, the Canadian Alliance for the Canadian Atherosclerosis Imaging Network, etc. And all of them were really outgrowths because we had these data sets, and we were credible contributors to lead and also to be part of larger studies. So it was fun digging up. Dave Callan is now a pediatric neurologist at McMaster, and he was doing tracing experiments that were mapping out the signature areas in the limbic system of Alzheimer's disease way before automatic methods made it possible to do in an instant. So there again, maybe staff. And the other thing is I realized how important administrative skills are when you're trying to do complex, run a complex set of studies and units, especially when we got into Health Canada approved studies, which are a whole other kettle of fish in terms of the complexity and how you have to kind of change things or not change things with notifications and amendments and all kinds of trouble for the REB and for time it takes to make things happen. So I do have some conflicts of interest in what I'm going to talk about. There's nothing really relevant to what I'm going to talk about, but this is now getting into the standard type of presentation. I've been participating in pharma trials for many, many years. Again, part of my autobiography is that it was the pharma trials in the 1990s that allowed us to recruit efficiently to the cholinesterase inhibitor studies that were underway. And we made a little extra cash because we got people in quite well. And the little extra cash allowed us to buy a computer, and then maybe even have a graduate student. And one of those graduate students I showed in the previous slide is Brian Buck, who started off doing neuropsych assessments in the Sunnybrook study. Eventually, he became a stroke neurologist, and he's now a stroke lead at University of Alberta. So it was a clinical trial that helped kind of lubricate us in getting extra things that wouldn't be paid for by a grant. These are some of my lectureships, some consulting, and then the grant fundings, and very grateful for salary support that I've had through the Sunnybrook Research Institute, the Department of Medicine, and now more recently the Center for Brain Resilience and Recovery. So what I want to do is I want to show how research embedded in care gave insight into vascular pathologies, which are intertwined in the development of cognitive decline in aging and in Alzheimer's disease. I think that was what we discovered very early on, has become sort of our brand, and that now the rest of the world is beginning to understand and take on with much more interest in this. They call it vascular contributions to dementia, but it's really what we were seeing just when we were observing patients in a clinic with the protocols we had. So what I'm going to talk about is that it may be understood when you have focal stroke that affects language area or visual spatial area, but it's different when you have just like what looks like a diffuse atrophy process going on, and that's something that we began to understand better because we could use SPECT scanning, which allowed us to see functional hyperperfusion, which actually showed that there were focal abnormalities in these different disease states that led us to be able to diagnose them and validate, when people eventually died and we did the postmortems. So part of what we discovered is location does matter even in these so-called diffuse diseases because they disrupt networks, and even small vessel disease has some peculiar effects depending on where it happens to be located. So the idea that the brain's vasculature might be important to cognition seemed kind of not in vogue in the 1990s when everyone was so excited to have discovered these proteins called amyloid and tau, and Alzheimer's disease was going to be conquerable by just blocking tau or stopping blocking amyloid or stopping tau, the two pillars of the disease. And the vasculature was sort of forgotten. This is when you inject a person post-mortem with a silicon-colored kind of rubbery substance, so it actually shows all the vasculature. And this represents about 400 miles of vasculature, according to some of my colleagues, basic science colleagues, including Beta Zlokovic, who's a world leader in understanding experimental biology of the neurovascular unit. So how could you not consider the vasculature when you're talking about the brain? And in fact, just like I've been describing family, my own family, my academic family, my institutional family, also in the University of Toronto, when you want to understand what's happening in the brain, you have to understand there's a family of nerve cells that are working together, a family of brain cells that are working together to keep everybody okay. Just like, as we know only too well in this pandemic, you need food and water. You need things to do, things to protect you, and you need things that are interesting to do. So you have here, the vasculature is actually the center of this little universe, this family, because it's providing oxygen and glucose to the cells that need that, especially the neurons and glia. It's surrounded by a layer of cells called the endothelium that's a very active substance that actually secretes things good and bad, and also can allow the arterioles to expand or contract. So smooth muscle will respond to that, and when you need more blood supply, then the vessel opens more, and if you want it less, you constrict it. So that's, you know, exquisitely controlling the flow of the nutrients. The astrocytes have to do with homeostasis and sort of protection, as do the cells here, the perivascular cells that are like the guard cells. Maybe it's kind of the mother and the father, if you want that analogy. The guard cells are fighting off attacks and being there to sort of guard the family. All of which is to protect this little neuron. Here's a neuron end foot, an axon projection. So this is a family of units with many different functions going on. And once you have that sort of concept, you understand, first of all, you need a village to bring up a child. You need a larger village to do research. You need an institutional support system to be able to do work that's recognized internationally. And all of that comes down to being lucky enough to have a good family to live with at home, and a family that supports you when you're in the academic setting. And I think mentorship has been one of my main goals in life, and I've been rewarded many-fold over from the wonderful, much smarter people that I've had the privilege to supervise and help grow. So let me just move on to why this is so important in understanding Alzheimer's disease, because there is this sort of axis of heterogeneity, which means everything is different, everybody's a bit different. You have to try to sort out who's who and what's what and what may be the best treatment. Eventually we're moving to what we call precision medicine. And this is this idea that you can have co-pathologies like white matter disease and Lewy bodies, which is the Parkinson's occlusions. You can have genetic factors, something called apolipoprotein A4, which gives you a great risk of developing Alzheimer's disease in older life. You can have age of onset, which also can have some genetic aspects to it, especially if you mutation. So sometimes there are mutations that means that you'll get sick in your 20s and 30s and you'll be dead in your 40s with Alzheimer's disease. It's a lethal disease at a young age. But also it can start without a mutation. We don't fully understand it in what's called early onset Alzheimer's disease, where the person may have visual spatial problems. It's called posterior cortical atrophy sometimes when it's bilateral. Or they may have language problems. This is called logopedic language variants, or logopenic Alzheimer's dementia. That often occurs at older age as well, but you can have language variants, you can have visual spatial variants. And the typical variant is a person who just presents with severe short-term memory problems. But even that is called temporal variant when the person has primarily memory problems and they're old, usually in their 80s and beyond, because there's another protein now that we know is contributing to that. Often it's called TDP43. You know you're in trouble when they just give a number to it because they don't understand it yet, but they can stain for it. And here's another important message. And so this is meant to be educational for those who may not understand these diseases so well. This is from a pathology series that was done in Chicago. And they had, on the left, they showed people with normal cognition, no cognitive impairment, mild cognitive impairment, which means that you've got objective findings, but you're still functioning well. And at probable Alzheimer's disease, in this case, they were looking for, which means you have dementia, the Alzheimer's dementia, where you are beginning to need help to manage in day-to-day life. So you've got multiple deficits, usually including memory, and you can't perform all your activities of daily living. So in that context, you will note that people who are thought in life to have Alzheimer's disease, only a fairly small number, it's about 10%, have only the Alzheimer pathology of amyloid and tau. And if you look, you can see that the other most common accompanying pathology has got a V, which means vascular. So Alzheimer's and vascular, Alzheimer, vascular, and other, which could be the protein of Parkinson's, the protein of this TDP43, for example, or even sometimes of other types of dementia, like frontotemporal dementia. And so you really do have to understand the vasculature and treat the vasculature and protect the vasculature to mitigate Alzheimer's disease. Likewise, V predominates in the mild cognitive impairment state and in the non-impairment state. And so this, to me, this has been replicated a number of times. This is one of the most convincing examples of this, which underlines to me that you really have to embrace the entire condition. So this got us going on this idea of a personalized imaging pipeline in the Sunnybrook Dementia Study. We took all comers, but they had to speak English well enough to participate in cognitive testing. They had to have about grade 8 education, because we were doing assessments that required some degree of education. They couldn't have other conditions like kind of chemo fog or severe depression, but we tried to be pretty liberal in letting people in. And then, so it was not everybody, but it was anybody who was sort of willing to participate in this study. And then they were offered cognitive testing, the specific protocol for imaging, and these other questionnaires that we would do with them, and eventually blood tests, which led us to be able to do some of their genetics. And this is just showing you the numbers we had mostly were Alzheimer's and mild cognitive impairment, frontotemporal dementia, vascular, Lewy body, and normal controls. And there are a whole bunch of other spin-off studies at 3T that we've done over the years, making the total number closer to 1,600 now that we have data on. The sequences are shown here. Again, this is rather technical, but the 3DT1 allows you to do a three-dimensional construction of the brain. That's now allowing us to generate the volume of a hippocampus that we used to have to trace lovingly, 6 hours per brain. Now it's done in about 20 seconds. But that's because of the ground truth that we help to contribute by doing this very, very labor-intensive frontline work. The PDT2, the proton density T2 allowed us to see something called perivascular spaces. The flare brings out this white matter disease, and the gradient echo brings out the so-called microbleeds. I'm just going to run you through the way our pipeline works. So now we can take pretty well all comers. We can correct, because of this, we also see the edge of the brain properly. We can correct for the head, for the outside of the brain and the inside of the brain. We can use some of the newer tools, enhancing them so we can measure cortical thickness, gray matter volume, regionally, in a way that's really unique still to us. But we're trying to make the pipelines available as apps so other people can use them. And this has been Joel Ramirez's work, as well as other individuals. And now it's been sort of machine learned and used to speed up things so that a brain volume and ventricular size can be generated in a matter of minutes. Now, this shows you, if you just do certain kind of imaging, which most of the other world does, rest of the world does, which is called a flare, which came along in the 2000s, I guess, you can actually miss important lesions deep in the brain. Here, the thalamus with a red arrow. I guess you see my arrow, where the red arrow is pointing to, these are little tiny lesions in the thalamus. And on the flare on the right, you don't see them at all. So if you don't do the PD2 or you don't have a T1 at the same time, you would miss those. And that's a very important point. When you extract the white matter disease, you can get three-dimensional views of it. It gives you a sense of this. And we also made the white matter hyperintensities be measured in relation to if they were actually by the ventricles or away from the ventricles. That's what we call deep, and we call periventricular. Some of the other techniques that are very popular around the world, if you don't take this all into account, end up missing out sections of the brain shown here. This is something called pre-surgery. It's widely used, and you can see that it just under erodes in this case, and sometimes it over erodes the brain, shown here, so that you really aren't getting a proper view of the brain. And they use the brain as the denominator, whereas we use the intracranial space, which doesn't change with age, and at least older age. So that is the denominator that we use so that we can know differences between men and women and big heads and different shapes of heads. So with that background, let me just run you through some of the faces of small vessel disease. These are, this would be on the extreme left, it would be an acute subcortical stroke. It looks hyperintense. If you look over where it says lacunes, that stroke might cavitate or it might not. It becomes a hole, and then you can see it as a hole on the T1. The white matter hyperintensities, and it's under WMH. You can see there's like fluffy patches. The lacunes are holes. The perivascular spaces, they're little kind of streaky lines that you can see. The little black dots are the microbleeds. And just recently at 3T, we can now see what are called microinfarcts, which are just like 3 millimeters. And you can see them if you look really carefully. And it turns out that they're actually quite important. You never imagined you could see this at 3T, but you can. Here's an example of a small lesion that can have a big cognitive impact. It's a lesion in the left, where the red area is the left anterior nucleus of the thalamus. And the rotating head just shows how small it is, but it's a key hub that's connecting that part of the brain to the fornix, which is where the memory area is, the hippocampus, to the language area, Broca's area, where you produce speech, and to the left frontal region, which is important for abstract thought. So in one fell swoop, a person can be able to speak properly, to think abstractly, and to remember verbal information. So it's very, very important strategic lesion. Example here of a gentleman who presented with confusion to the emergency room. A CT was negative. We actually didn't pick it up early because often the old CTs weren't so good at that. They are better now. But he had short-term memory loss, word finding difficulty, and problems with kind of judgment, and this abstract thinking in a way that made him no longer able to function. And what we were able to show is that he had a large enough lesion in his left anterior thalamus that he was made demented in a matter of minutes from this one fell swoop. This is a strategic cause of dementia related to stroke. Now, there was a young guy called Rick Swartz who was learning how to do, or thought it was important to do, simultaneous assessment of the atrophy and the white matter disease and the focal infarcts, which was so novel at the time that when he tried to publish it, people didn't believe him. We were just talking earlier today until 4 or 5 years later. By that time, he was in medical school and probably in residency. And a special issue of Stroke came out saying they wanted, to do some emphasis on small vessel disease. So I told him to maybe put this paper that had been lying there for a few years, waiting to be revised. I said, why don't you just try to send it in and see, update it a bit, see what happens. And he did that on a weekend. I guess, Rick, there wasn't much update. That was 2008 or something. And the review that came back, I told him to frame it, was bravo. That was the entire critique. Whereas people see, they criticize this or criticize that because people are beginning to get the idea that maybe this was important. So he showed that there's a kind of a size of injury that seems to be associated with the symptomatic decline on the left. It's around 55 cubic millimeters. On the right, where it has more to do with visual spatial functioning, you needed, you didn't end up having dementia, but you had cognitive impairment. So that was kind of interesting. And then he also showed that even little hyperintensities in the thalamus, independently, along with other things, correlated with executive and memory problems. It was really showing that the thalamus is actually very, very important. And I think Rick probably still has PTSD from the day he came into his thesis committee. We had an expert, an anatomist at the University of Toronto, who said, okay, Rick, draw me all the nuclei of the thalamus. He wasn't quite prepared for that. Peter's laughing, my brother, because he knows that you have to know all the nuclei of the thalamus when you're doing surgery. Anyway, he sort of did it. It wasn't too bad. But then he had it memorized, kept waiting for her to ask again, and she never did. But he knows all the nuclei of the thalamus because they got imprinted along with that embarrassment, because he sort of got a few of them, right? Moving on then to arteriolar disease. Here you can see on the left, it's usually thickening of the arterial wall. It's called hyalinosis, which leads it to stop providing blood flow, so you get a hole in the brain. You can also, in the lower left, you can see a straight artery that's coming from the brain down towards the ventricles, if you're thinking of the brain in 3D. And to the left of that, you can see that sort of ugly looking, bumpy black stuff. That's actually amyloid going along the perivascular space of that artery. And you can imagine that that is a sick artery. It's not doing well. It can stop blood flow from being adequate. It can even bleed. It can be the cause of bleeding. And so it's always important to remember that Alzheimer's disease pathology, amyloid, can be the cause of stroke. It can be the cause of fatal stroke. Now on the right is arteriolar disease. It's actually a little bit different. This is looking at the length. The upper right shows you the actual length of an artery, and it starts as you get older to remodel and become curly. On the lower panel on the right, you can see a straight looking artery, which hopefully most people on this, maybe Andy and I, not so much, but probably have pretty straight looking arteries. And then as you get older by each decade, they start to remodel. And when you get to the right two panels, there's actually no blood flow anymore in those arteries from remodeling. The important point is this can happen with aging and the absence of any known vascular risk factors. So just aging can contribute to this white matter disease. And we've seen this, people with severe white matter disease who actually have no vascular risk factors. And we suspect it's something like this that's contributing to the lack of blood flow that hurts the veins, which we'll talk about next. This is an example of what the perivascular spaces look like. In the pictures down below, you can see little streaks, and you can also see little holes deep in the middle part of the brain with the arrows. And we developed automatic ways to measure this where everybody else was counting them. So we had continuous measures, and we could look at the relationships. And we showed that when it was in the basal ganglia deep in the brain, like where the arrows are in the middle, 73-year-old men with vascular dementia, they were often associated with amyloid angiopathy. They were associated with high blood pressure. And when they were in the outer areas, the so-called lobar regions of the brain, shown on the right as these little kind of linear bright spots, they were associated with amyloid angiopathy. We also showed that after stroke, when they're enlarged, these were thought to be enlarged spaces because they're pretty small otherwise. When they're enlarged, they were associated with disordered sleep after stroke, including sleep apnea and restless periodic limb movements and so on. And that was because we were able to compare quantitatively some aspects of sleep with these continuous measures. This just shows you the arrows are pointing to the little microinfarcts. You can see how hard those are to detect, but it turns out that they are important, and they show up in memory clinics being associated with cognitive impairment. Now, these were completely off the radar. Now they're on our radar, but they're still very difficult to find. You have to be an excellent radiologist with focused attention. We've had the benefit of Dr. Fucheng Gao for all these years. And he does all our incidental findings in these studies, and he's also mapped out the venous disease, but he's the one who finds all these infarcts for us. He shows you microbleeds deep. Those are associated with hypertension more often. Microinfarcts lobar, microbleeds, again, black on a certain kind of scan. And this is a 69-year-old lawyer presented with confusion. He was bumping into his desk, and people thought he was drunk or something. He was taken to the emergency room, and this is what his CT showed. When you have a hemorrhage, you can usually see it on a CT. And sure enough, there was a large white blob which was an acute cerebral hemorrhage on the left side of the brain, because the left is always on the right side of the scan by radiological convention. And he had a full-blown set of symptoms shown here. Well, I'll talk about that in a moment. These are the microbleeds that we saw in him when we did the gradient echo a couple of weeks later after he recovered somewhat. And you can see where his hemorrhage was, where those black blotches are. That's what's left of the hemorrhage. It's being cleaned up by the brain. But around, in other parts of the brain, there are little dots in the areas of blackness, meaning that he had microbleeds in the lobar regions of the brain. He had no history of hypertension. And his syndrome, I guess I cut some slides out. His syndrome was that he had profound transcortical sensory aphasia. That is, he had kind of speech that was not always meaningful. He had problems, some problems with he had word finding difficulties, some problems in his comprehension of speech. He also had apraxia. He had a field defect, and he was never really able to return to work, although he lived for about 9 more years. At around 6 years, he started to show dementia in the sense of memory loss and had problems with his day-to-day functions. When he died a couple of years later, he had a combination of these cerebral hemorrhages remaining and a pretty advanced stage of Alzheimer's disease. So this can often tell you that the person may also be harboring Alzheimer's disease. This is showing you that you can see here some white stuff in relation to clinical trials. And I bring it up because we are now in an era that's becoming really, really interesting. We have antibodies to amyloid. We have antibodies to tau. Fast forward from the 1990s to now. But they can be associated with some side effects that are thought to be the amyloid plaques being sort of made more able to be removed from the brain along the vessels, and sometimes it just overwhelms the vessels and you get this sort of edema. It's thought to be vasogenic edema, and that can be a limiting factor in the use of these treatments. We're actively involved in trials right now and have some people that have had to stop their treatments for a while to see if that will resolve. If it does, they may be able to continue. If it doesn't, they may not be able to tolerate the drugs. And also, if they have more than 5 microbleeds to start with, then they would not be allowed to do a trial, because you can also have hemorrhagic microbleeds from removing the amyloid, and blood escapes as it's going along the vessels. It just sort of shows you the picture. And this person, it did eventually resolve. So let's talk about white matter hyperintensities, white matter disease. And I wanted Andy to see this. Andy, this was a scan. This was a scan we did at the 0.15 T scanner. And these were the rims and the caps, which we call UBOs, right? Unidentified bright objects, back in 1988. So we've been sort of fascinated by these ever since. I just point out one of the arrows to the right at the bottom there. You can see a little linear streak. And so I'm going to go on to say that now I think we have been able to better understand what's going on in white matter disease, the so-called leukoairiosis, which means rarefaction of the white matter. It's important stuff, because longitudinal studies have shown if you have this white matter hyperintensity, you're at increased risk of stroke, cognitive decline, dementia, and death. In fact, in one study, if you had moderate white matter disease, you had 3 times the risk of stroke, 2 times the risk of dementia, 2 times the risk of death. And you were also likely to have poor overall global cognitive functioning, and you were slow in speed of processing. Also, it means better, like worse outcome after you have a stroke. You don't recover as well. So this is what it looks like, and this sort of shows you different volumes. And don't worry, I'll be getting to toward the end soon. But there are people in the audience who hopefully are still with me who may not be aware of what we're doing all the time when we're doing the scans and measuring things. So these are on the left side are scores. It's called the Physica score, which allows us to do a quick read now. And I notice that our radiologists are now giving us that score when they do the assessment. They also give us a little rating on the hippocampal atrophy, which is handy. But we can actually measure it pretty quickly now. And this shows you what 5 cubic centimeters looks like, 10 cubic centimeters, 25 and 50. It's thought around 10 cubic centimeters, which is this Physica score 2, shown in the second row. It starts to be associated with some subtle cognitive problems. By the time you've got 25 or 50, you're certainly slower in information processing and have some of these other executive deficits I mentioned. This is a woman who came to us after she'd been in the NACIT trial. Peter will remember the NACIT trial. So would Andy, because it was a trial of endarterectomy in the carotid arteries. And she was randomized, Barnett was in charge of, she was randomized to the medical, she had a TIA that caused some left-sided weakness, and she was randomized to medical treatment. And over the next few years, she started to have cognitive complaints. And we saw her in our clinic, and at that time, this is what year 5 after she had the endarterectomy, she had quite a lot of white matter hyperintensity shown there. And we followed her for a few years. And this was closer to around the time that she died, which was about 8 years later. Now it was 71 cubic centimeters. And so this is kind of interesting. Andy will be interested in this because he was very interested in white matter disease and in pathology, as well as contributed hugely to our understanding of frontotemporal dementia and the progressive aphasias. So Andy, when the autopsy was performed and the neuropath was done, a very good neuropathologist didn't make any mention of anything around the ventricles. And we said, well, there's got to be something wrong there. Look at the scan. And so he went back and he said, okay, well, I'll do some more. He said, this is a person who's demented. We look at the cortex. We don't look at the white matter. It's the cortex that matters, right? So apparently it was not standard to look at the periventricular region at all. The only person who did that regularly was a guy called Harry Vinters, who went to California, who you will remember, I think, Andy, as well. He was a Canadian and has always been interested in vascular cognitive impairment. So anyway, what it showed in the end, when he went back and stained, is he found that there seemed to be some funny thing going on in the glial cells, those protection cells that I mentioned, the astroglial cells, and they were ballooned and they had beads and they looked sick, like they were trying to imbibe fluid or something. And we reported that. We didn't really know what was going on then because we hadn't figured out that one of the things you have to do when you're doing vascular pathologies is you have to do something called trichrome staining. So this is what she showed. She showed this kind of these, to the lower right, you can see these beaded abnormal astrocytes. You have to take my word for it. They look very abnormal. And interestingly, that had been described by Alzheimer in like 1911. And he thought it was associated with hardening of the arteries and with kind of ischemia. He's probably right. But here is what we think is the clue. This was the clue, Andy, that I've been tracking since those UBOs in the 1980s. If you look at the colocalization of the white matter hypertensities in the deep penetrating venular system, you can see that they are exactly colocalized. And this is the beautiful work of Fu-Chung Gao, and we're just in the process of getting this finally ready for publication. He actually traced the venular system in this particular individual, showing it in 3D actually. But you can see that they are colocalized. So maybe there's something to do with the veins going on here. Nowadays, you can actually see this quite beautifully. This is a susceptibility weighted imaging acquisition at 3T, and you can see these veins. And this is the perivenous white matter disease. You can actually see the veins. You can even see that they go right to the cortex. Some of them are transcerebral, others are just receiving the input from the arterioles. And you can see various other things here. You can see the even focal ones. I think I've maybe got the wrong protocol here. Anyway, they co-localize, and the important point is at post-mortem, what you see is they're associated with hardening of the venules. So you have to do something called trichrope staining in order to see this. Sometimes they're completely occluded, sometimes they're partially occluded. What we found in an important study with Dr. Keith, who was our neuropathologist, is that the white matter hyperintensities are associated with thickening of the large, deep medullary veins. The demyelination that also occurs when you have white matter hyperintensities contribute, but not as much as the veins. So you have to actually stain with something called trichrome stain to see this. And so far as we can tell, nobody ever does that. Not even the important series in the Chicago Aging Study and all of the centers in the US, they're starting to. And I'm happy to say that there is a group in Oregon that had a similar protocol, because you have to have the imaging prior to death to be able to correlate it with the hyperintensities. And they have just replicated our finding using actually a area tracing of the wall and looking at perivascular spaces in the nearby arterioles. And a very elegant study, I think, is going to be forthcoming from their replication. I'm going to just quickly go through the cholinergic projections, because again, this is an idea that I put to this young guy, Rick Swartz, and said, you know, maybe we should look at this white matter disease in relation to these trajectories of the cholinergic pathways. And at first he didn't pay too much attention to that, but then he thought maybe it'd be a good thing to actually come up with a little scale. And I think you actually presented it at a meeting which DeCarly was at, and pretty soon I think some of that was taken up by him. But anyway, the cholinergic deficits that we see in the brain are associated with something called the nucleus basalis of Maynard. And you can see that it's not just in Alzheimer's disease, but also in patients with Parkinson's, Lewy body disease as well, compared to controls. And so this little scale, when we started to apply it, allowed us to see that this was a strategic location. If you injured pathways that involve these projections, but especially in a part of the brain called the external capsule, then you could show that it's actually associated with sort of different patterns. I won't go into this because I'm running out of time, but here it shows that we've automatized measuring that now so that we can do it quickly without having to do the rating scale. And we've shown that white matter hyperintensities in those pathways are associated with poorer memory function and actually reduction in hippocampal volume. So it's always thought that the white matter disease didn't have anything to do with the memory area, but it actually does. And also in a post-stroke sample, if you had a stroke in that area of the very left part of the lower level where it's green, if you had a stroke involving that area, or you had white matter disease, then you had problems in what's called the Trails B test, where you have to have working memory and keep shifting from one movement to the other. You have to kind of alternate between numbers and letters, joining dots on a page. It's a very brief, simple test, but extremely telling. It is used actually for driver assessments, because it means you have to keep switching attention and you have to kind of remember what you've just been doing. And it showed that involvement in those pathways specifically associated with that executive function. Whereas if you just looked at the total amount of white matter disease or the total size of the stroke, it tended to be more affecting and associated with speed of processing. We've also shown that if you have remodeling of the carotid artery, which just happens to bifurcate at the part of the brain that contains this important nucleus where all the cholinergic neurons come from, you can actually show that it makes it shrink and that it actually correlates with atrophy of this important called basal nucleus of the forebrain, That was done in one sample, and we replicated another sample and showed that it correlated with executive function. So remodeling of your large artery in that particular area is associated, after you account for other factors shown here, is actually associated with cognitive injury or cognitive decline. And it's thought that's because you're probably pounding that area, or maybe there's ischemic injury to it. And it's a kind of a novel insight into how large vessel disease at a strategic area can also impact. So I think there are other studies now that have shown, even in the ONDRI study, that maybe the first thing that starts to happen in the dementia syndrome with Alzheimer's disease is shown on the left, the brown line on the left, which to everybody's surprise actually referred to vascular changes, as opposed to the accumulation of amyloid and tau, and then the neurodegeneration. This was actually very shocking. A lot of the kind of Alzheimer community just thought it can't be right. But there's a reason that this may be happening. It's because the areas of the brain that are in what's called watersheds, where they're underperfused, are also in the areas of the brain that are very, there's association areas that are very linked to neuroplasticity, where you're bringing together many inputs to guide action at the back of the brain, the front of the brain, the dorsolateral part of the brain, and sort of in the mid areas. And so where the vascular supply is most problematic is also the part of the brain that's most neuroplastic. And so it may be that these two pathologies converge to make those areas more important. And these are considered the signature areas of Alzheimer's disease, but they may really be signature areas of hyperperfusion related to the watershed. So small vessel disease is ubiquitous. It can be overt or covert infarcts. It can be elucidated by computational methods, and we can't wait to get our new CFI funded 7T MRI, because now we can see those vessels. It's like having a low field magnet, so we're very eager to have that coming into play. Perivascular spaces are very important, and the venular system has been totally ignored, and it really can't be anymore. But you just got to state it if you're doing pathology, and you have to be aware of it when you pay attention to where pathology is in the white matter hyperintensities. And we look forward in the future to doing more physiological measures of blood flow and cerebrovascular reactivity and functional connectivity, which our group is now actively pursuing. And so I think in the sociology of science, you need interdisciplinary teams in care as well as in science. And we've been able to spin off some of our own projects, but also it's allowed us to be a hub in many networks. And I have to say that underlining all of this, because we, generally speaking, are not as well funded as many other countries for the work that we do, and we don't get sort of salary support from those fundings. The importance of philanthropy to enable and to fuel and accelerate these activities has been huge in our experience. I'm very grateful to Linda Campbell Foundation for the money she gave several years ago to accelerate our pipelines, and also especially appreciative of the anonymous gift from the Nikita Foundation. They insisted it be called after me. I find that embarrassing, but anyway, it's the Dr. Sandra Black Center for Brain Resilience and Recovery. And we just had one of our first meetings as that's become active recently today. And it was just unbelievable, the amount of exciting science going on with all of the individuals who are being funded by that wonderful lead gift. So I just want to, again, acknowledge the support from many trainees and colleagues, and once again, circle back to where it all was allowed to happen. Thank you very much, everybody, and thank you for this wonderful honor. I really, really appreciate it. And if anybody has any questions, I don't know how you want to handle this, Rick. Please go ahead. These are some of our various references, and again, all of the various logos, including the Ontario Neurodegeneration Research Initiative, which is basically Rick and Mario, Ontario Brain Institute. I didn't give much emphasis to the genetic information that we're working on. That's really Mario's area of expertise. We're finally harvesting that. And also he's our expert sort of in movement disorders and frontotemporal dementia, and is leading international groups in that domain. So I think we've got a lot of angles covered, and I'm very, very proud of those I've had the ability and the opportunity to mentor over the years. And I was really touched by this leadership award, this Lifetime Achievement Award, and to be following in the footsteps of none other than Andy Curtez,